Yeah, we now have an example, the first major scientific discovery using AI. And there's a researcher called Paul Robbins that we have here. And he attended our conference, or rather the Nordic Aging Society's conference at Karolinska Institute yesterday. And I really wanted to have him here because he's working on something called senescent cells, which are sometimes called zombie cells. And I just had to put this illustration here because it's funny. Now, he was part of the group of scientists who actually discovered and characterized, or at least he's worked very closely with senescent cells. And I think this has the potential to be one of the next very big breakthroughs that we can use AI to have a meaningful impact on our health. So he couldn't be with us here today. He flew back to Minnesota, where he's the professor of biochemistry and molecular biology and biophysics. But we are lucky enough to have a small video. I asked him, could you please record something to our conference attendees? And he was happy to do so. So we will now watch a very short video, I think it's like 4 minutes, where he describes who he is, what he works on, and he has a quite exciting new large project that the NIH, the National Institutes of Health, has sponsored. And I think this is very significant. And I won't spoil it, but we will now look at the video. And
hello, my name is Paul Robbins. I'm the co-director of the Institute of the Biology of Aging and Metabolism at the University of Minnesota, which I co-direct with Laura Niederhofer. I'm actually talking today from a hotel room in Stockholm. I'm attending the Nordic Aging Society meeting at the Karolinska, but unable to make it on Wednesday to your event, and I really apologize for that. So my lab for the last 10 years has been working on something called cellular senescence. And this is a cell fate, so cells undergo this process in response to damage. And this could be genotoxic damage, oxidative stress, it could be activation of oncogenes that cause cells start to grow. It could be inflammatory stress, exposed to radiation, UV, a variety of things, including tobacco smoke and other environmental factors that are in your diet. And what happens is cells become damaged. It's not a good thing. So these cells exit the cell cycle so they can't keep proliferating. And they start to secrete what's called an inflammatory cascade, or what's called SASP, for senescence-associated secretory phenotype. And this SASP allows immune cells to enter the tissues where these senescent cells are arising and clear them through an immune-mediated clearance process. And so this is thought to have evolved as a tumor suppressor mechanism. So if cells are damaged, acquire mutation, and start to replicate, this process is turned on. These inflammatory cytokines and proteases that degrade the tissue, which allows these immune cells to invade into these tissues, to clear them in order to reduce the risk of cancer. However, as we age, our immune system starts to fail and doesn't clear these damaged cells. And so they start to accumulate, and they can cause chronic inflammation, loss of tissue homeostasis, and can contribute to driving aging, frailty, and age-related diseases. So my lab has been working on approaches to actually get rid of these senescent cells. We're developing drugs called senolytics, which can target specific pathways which are activated in senescent cells that lead to specific killing of these cells, similar to what you would do to kill a tumor cell. And I'm also part of a consortium. It's a large consortium funded by NIH. Comes from the Common Fund from the NIH director of about $190 million over the next 5 years. And the goal is to actually identify, characterize, and spatially map these senescent cells with healthy human aging and healthy mouse aging. And as we can identify and map these cells, it will give us clues to the role they play in human disease, as well as mouse disease, and will allow us to develop ways to target the correct senescent cell types that contribute to driving aging and pathologies. But as part of this effort, we're using a variety of omics methods, including spatial transcriptomics, single-cell RNA-seq, proteomics, including CyTOF, flow cytometry, lipidomics, metabolomics, to try to characterize these cells and then map where they exist in the liver, in the kidney, in the brain, muscle, adipose tissue, and at other sites. And as part of this effort, we're generating huge data sets. And we need to mine these data sets to identify these senescent cells, which unfortunately are very heterogeneous. And so we coined the term phenotype to kind of define the variety of different types of senescent cells that we appear to be identifying in different tissues. And so as part of this consortium, these data sets are being uploaded at the website called senetconsortium.org. There will be over 1,000 data sets there, very large data sets. And what we need help with is actually mining these data sets to identify these rare senescent cells that actually contribute to driving disease and pathology. So if you're interested, I encourage your group to be thinking about these approaches and seeing about ways you can potentially integrate with this SENET consortium, because I think over the next 5 to 10 years, this characterization of senescent cells is going to lead to new therapies that allow us to live healthier, if not longer, but live healthier for longer, which is the goal which we're all striving for. So again, I apologize that I'm not there. Hopefully this short video has given you some food for thought about ways that you could apply AI to actually understanding and characterizing and mapping these rare cell types that arise with aging and disease and help move the field forward. So again, thank you very much. I look forward to any feedback that I might get. Thank you very much.
Right. Woo. Paul Robbins, everyone. All right, and to reiterate, why is this important? Well, senescent cells, if you clear them in model organisms, we extend the healthy lifespan. And he just announced that NIH is funding a $190 million initiatives to go out and find these senescent cells, which they're not the same, but they are everywhere in our bodies. So this is really significant, and we can use the AI tools to then to take away these senescent cells. So this will have huge implications. Okay.